right, let's tackle part two here, which is going to end up being the Calvin cycle. So here's the little refresher roadmap diagram. So we went through the light reactions. We converted sunlight and water into oxygen, which is given off for all of us to breathe, into ATP and NADPH, light into chemical energy. Chemical energy is now going to power the Calvin cycle. So let's go build some sugar. So remember what it means to be a plant. You have to produce all your own organic molecules, right? You are not a heterotroph. You have to make things. So you're going to store the chemical energy, the ATP produced, from light reactions in a more stable form, right? ATP doesn't stick around very long. So if we can convert it into sugar, or sorry, if plants can convert it into sugar, then they can kind of have more of that energy on call when they need it, because you're going to store it in the bonds of the glucose, right? And so we can save it up for a rainy day. So there's the equation at the bottom with carbon dioxide, water, and light energy going in, and then glucose and oxygen coming out. So the light reactions, we converted solar energy into chemical. We have ATP and NADPH. What can we do now? We can build stuff, which is really what photosynthesis means, right? You're building stuff from light. You're building all of that from sunlight. So it's really sunlight that's driving the building of glucose, right? So, which prompted one of my former students that said carbs come from the sun. Kind of right, because it's the beginning of the food chain, right? So, want to make sugar. We want to make C6H12O6, but what do we make it from? What's available? CO2 is available, right? Because we give it to them through respiration. This is the link between the two, or one of the links between the two things. So we're going to take CO2 and we're going to keep adding to it, and we're going to build it up into a glucose, into a sugar. Okay, remember, respiration took glucose and stripped it all down until all we had left was CO2 to exhale. So obviously just the reverse, right? So we call this carbon fixation because we're going from 1 carbon to 6 carbons. We're going to fix carbon onto the CO2 and keep adding to it. And we're going to spend some NADPH as well. And that's going to be what's reducing the CO2. So we're going to give it some hydrogens, right? Some electrons to build up that sugar. So CO2 has very little chemical energy, right? Fully oxidized, but sugar contains a lot of chemical energy, highly reduced. So we're going to try to make that happen. So it's an endergonic process. We're putting in a lot of energy. And obviously all of these are really, small steps, just running in reverse of respiration in order to build this up, all by specific enzymes that make this happen. So here's our little diagram again, and as far as our summary goes, we're going to have the Calvin cycle, and it's going to take place in the stroma, out in the space of the chloroplast, and you're going to need that ATP and NADPH to drive the sugar building reactions. So here is the Calvin cycle, and it can get a bit complicated, so you're not going to have to know every little bitty thing about the Calvin cycle. Big ideas here, okay? So the first thing is, at the end of the cycle, you're going to have these 5 carbon molecules, okay? And then CO2 is going to come in, bringing the extra carbon, or the last carbon, to fill out kind of these 6 carbon molecules. And there's an enzyme that's going to fix the CO2 to that 5 carbon molecule, and that enzyme is called Rubisco. Obviously, that's an abbreviation. There's 3 big long words there, but we call it Rubisco. And I'll come back to Rubisco in a little bit. So ribulose bisphosphate carboxylase. There you go for Rubisco. So you're going to take in some CO2, and now you have this 6 carbon molecule, right? 5 plus 1 is going to give you 6. So now you have a 6 carbon molecule. That was carbon fixation right there. Rubisco does the carbon fixation. And now we're going to have a little chemical rearrangement, and we're going to take 3 6 carbon molecules. We're going to turn those into 6 3 carbon molecules. So little chemical rearrangement, no big deal. Don't worry about the PGA. Don't worry about the names. Okay. We're going to spend some ATP now. We're going to spend some NADPH as we move around. And we're going to just be doing chemical rearrangements here. We're going to be adding things onto them. We're going to be adding on some hydrogens. rearranging some of the other molecules. Now we have something called G3P. Okay, G3P is the first building block for glucose, and so part of this is going to come out, right? So if you count over here, let me grab the pen. Over here we have 6, right? But if you look over here, 1, 2, 3, 4, 5, here's your 6th one, the G3P, and it's going to sneak out of the cycle. Okay, it's not going to keep going. So we sneak one out, and then we're going to have this process keep going. Okay, and it can make different types of sugars, potentially glucose, potentially something else. And those 5 carbon molecules are going to keep going. We're going to spend some more ATP, and we're going to regenerate the RUBP, the ribulose bisphosphate. We're going to regenerate that, and now we're back at the top of the circle, and in comes more carbon dioxide, and off we go again. So it's going to take some turns, multiple turns, to actually make a glucose. Okay, so the accounting gets a little weird. Okay, but as far as the Calvin cycle is concerned, the big things, your big takeaways, is that you have carbon dioxide coming in, that Rubisco is doing the carbon fixation, okay, in order to add the 1 carbon molecules to the 5 carbon molecules. That's just big picture right there. Carbon fixation takes place between CO2 and Rubisco, adds it to the other molecule. Don't really care about these names right here. This is all what I call chemical rearrangement of the Calvin cycle. You have the reduction going on down here. You're spending some of your NADPHs to then make G3P. It wouldn't be a bad idea to know G3P, just so that you know that that's the molecule that starts building the glucose, and then the rest of it just keeps cycling. And then this thing's going to spin a few times in order to make a glucose. It's not one turn, one glucose. The accounting is a little bit more difficult than that. Okay, but those are the big ideas that I'd like you to know. So G3P was actually in glycolysis, if you remember this step, the very first step of respiration. G3P was one of those in the steps of those 10 reactions of glycolysis. So what we're really doing here is we're running glycolysis backwards. Okay, we're taking CO2 and we're trying to build it up into a glucose, basically running a lot of the reactions of glycolysis backwards, because we're trying to get it up into the glucose. So we have to get some G3P in order to build it back. So the accounting, like I said, is a little weird. Not going to hold you to as much accounting here as I will with respiration. But basically, there's about 6 turns of the Calvin cycle to get you 1 glucose. Okay, so so you're going to need, 6 CO2s in there. You're going to need about 18 ATP and about 12 NADPH to make that happen. Okay, so it's a little bit more complicated. Not too big a deal there as far as the accounting is concerned. Photosynthesis summary: light reactions produced ATP and NADPH, consumed water, gave off oxygen as a byproduct. The Calvin cycle consumes the CO2, produces G3P, or the sugar that's going to build glucose. You regenerate ADP and NADP. I didn't talk about that yet, but if you look at the top right here, the top lines of this, after the Calvin cycle goes, you have these guys that get recycled back to the light reactions, right? So look at this circle right in here. Okay, as it just keeps going around and around, you have ATP and NADPH getting spent in the Calvin cycle, and then you have NADP and ADP going back so that they can make more. So these are your shuttle buses, just continuously running this inside circle between the two. Okay, so that's how you're getting a lot of your materials to do what you need to do for this. So sunlight comes in, water comes in, we make some ATP, we make some NADPH, oxygen's given off. Here's our equation. This is all light reactions, and it's going. Oops. One last thing, it's going to take place in the thylakoids, right? Thylakoid membrane for all those reactions. Then we go to the Calvin cycle, and we're going to spend some ATP and some NADPH to power the sugar building reactions. CO2 is going to get consumed. Sugars are going to get built. ADP and NADP are going to go back to the light reactions and get recycled so that we can make some more energy molecules. Okay, so it builds sugars, uses up ATP and NADPH and recycles ADP and NADP. Putting it all together, sunlight, water, in, ATP, NADPH, go over to the Kelvin, oxygen gets put out, CO2 gets consumed, gets fixed by Rubisco, and ADP and NADP get recycled, and whoops, and there you go. That's pretty much photosynthesis. So obviously we've seen this picture before, the energy cycle. This is the engine that's driving the planet, guys. I've said this before, any break in this cycle and we got problems, right? This is the great circle of life, Mufasa. Okay, this is how it goes. And so if we get any break in this, then we're going to be in trouble. Okay. So we have to hopefully make sure that this thing keeps going and our food webs and food chains keep going so that we get oxygen and they get CO2, and that's how the whole thing go. So supporting a biosphere, so on a global scale, it's the most important process for the continuation of life on Earth, photosynthesis is. Each year, photosynthesis captures 121 billion tons of CO2. Captures meaning from the atmosphere. It takes that many billion tons of CO2 out of the atmosphere and into the plant leaves to build sugars. Okay? And it synthesizes about 160 billion tons of carbohydrate for themselves, which actually then becomes sugar to feed all the things that feed off the plants. Okay? Heterotrophs are dependent on that food and on that raw material. So here's our summary with our equation up here. Where did the CO2 come from? Us, right? Respiration users give off CO2. Whoops, where did the CO2 go? It went into the Calvin cycle to build sugars. It became glucose, really. Where did the water come from? In the equation, you get that through the roots in the groundwater. Bring that in. Where'd the water go? Remember, it gets split, okay? And you end up with three different things. You end up with the protons, the electrons, and the oxygen that goes out. Energy came from the sun. Energy is used to convert into chemical energy. What will the sugar be used for? That's going to be energy for the plants, right? Cellular respiration will break that down and use energy for the plants. Where did the oxygen come from in the equation? That's when we split water. Where will the oxygen go? Out. What else is involved? NADP, NADPH, ADP, ATP, all those things in the center circles. Let's talk a little bit more about Rubisco, right? This is the enzyme that fixes carbon from the air, right? From the CO2 that we breathe out, you have the ribulose bisphosphate carboxylase, or Rubisco. Clearly the most important enzyme in the world. If you can't fix carbon, plants can't build sugar. If they can't build sugar, they don't have any energy, they die. Plants die, then all the food webs and food chains break up, right? So then also if they die, they're not putting oxygen in the atmosphere for us. So we have to have Rubisco. Fortunately, it's also the most abundant enzyme on the planet, okay? So we have that going for us. So there is some inefficiency of Rubisco. So what happens is Rubisco evolved in a low O2 environment. So which means it has a very high affinity for oxygen. It likes oxygen, but we don't want it to like oxygen. We want it to like carbon dioxide, right? Rubisco needs to fix carbon dioxide. We do not want it messing around with oxygen. We don't want it being a tramp and two-timing us, right? So if it doesn't bind CO2, then no Calvin cycle, no sugar, that's going to be a problem. So when oxygen concentration is high, Rubisco will try to bind with it. We don't want that. So O2 is a competitive substrate, right? It's competing for Rubisco. Not, because it's thinking about doing that. It just happens to bind really well with it. So if it's present, it'll happen. And so then you'll get the oxidation of the RUBP instead of the reduction of it. So now you will be breaking down the sugar. Well, we're trying to build the sugar, right? Calvin cycle is trying to build the sugar, not break it down. So the reverse effect will start happening if we have too much oxygen. So this process is called photorespiration. If we have too much oxygen around and it starts breaking sugars down instead of building them, Then you have photorespiration. And so um, there's a few things that goes on with photorespiration and how um, plants deal with this, right? They don't really want photorespiration happening. So when enough CO2 is available, Rubisco doesn't bind much oxygen. So when there's a lot of CO2, then it's not too big of a problem. And so plants have these structures called stomata. Okay, so you guys know that plants give off oxygen and they take in CO2. Well, where does that happen? If there's gas exchange, there have to be like pores, right, for the gases to get through, and that's what these stomata are. They're little holes, and they're actually in the underneath side of the leaves. Okay, so if you look at a leaf, you'll see like this microscope picture down here. Okay, you'll look at um, the underneath side of a leaf and you'll see stomata. That's what these things are right here. Okay, 2, 3. So these little holes right here and here, what looks like a little air pocket in there, or more, we look up here. This hole right here is the stomata. That's where the gas exchange happens. Okay, and so when the stomates are closed, they conserve water. They would want to do this if the day was really hot, really dry. You don't want the water to go out because you need water to do photosynthesis. And so they can close them up or they can open them up. Okay, and they do that with guard cells. So guard cells are right here, these two cells, or down here, it's these two cells, and they inflate or deflate with water, and they can either open or close that gap. And so if they gain water, then they open the stomata, and if they lose water, then the stomata close. Okay, so this is an adaptation living on land. Okay, but this also creates problems. So when stomata close, think about this. What's trying to go in and out, right? The oxygen is trying to come out and the CO2 is trying to go in. But if you close your stomata, neither is going to happen, right? So what builds up on the inside? Oxygen. Oxygen builds up on the inside, but that's bad because then you're going to have more photorespiration, right? Closed stomates lead to oxygen buildup from the light reactions. So light reactions are still happening, water's still getting split, oxygen's being produced, but it's not getting out of the plant. So now you have less CO2, more oxygen, and that causes more photorespiration. And now you're going to break sugars down instead of build them. So you don't want that. So plants have evolved ways to deal with hot, dry areas and still decrease their photorespiration to get around this problem. So they have different structures. One's called C3, there's another one called C4, and there's another one called CAM. And so C3 is normal. C3 is what we've talked about so far. Okay, carbon dioxide is fixed into the 3C molecule. Both stages occur simultaneously, meaning the light reactions in the Calvin cycle. And that's C3 plants are what we've been talking about the whole time. Okay, no adaptation for photorespiration. Some of it happens, but more photosynthesis happens, so it's not a huge deal. So if you look here, there's a C3 and a C4. So C4 plants are going to physically separate, they're going to physically separate the carbon fixation from the Calvin cycle. And if you look down here, this layer right here, this blue layer, which is called the bundle sheath, okay, the bundle sheath cells, it's way thicker than the C3, right? Way thicker over here than it is over here. And even the mesophyll cells look a little bit thicker than they do over here, and they look a little bit more abundant. So they are physically have a different structure that's going to help them with photorespiration. So let's take a look. So C4 plants are going to capture CO2, and the first step it's going to do before running the Calvin cycle is it's going to fix carbon with a different enzyme. It's not going to use Rubisco right now. It uses another enzyme called Pep Carboxylase. Okay, Pep Carboxylase is going to take the CO2 and put it into a 4-carbon storage compound. Okay, doesn't matter what the name is right now, but it's going to go into a 4C compound, which is kind of why we call these plants C4. Okay, and so adaptation to hot, dry climates and which means they have to close their stomata a lot so they don't lose water. Okay, and they get around this with different leaf anatomy. Sugarcane, corn, other grasses do this. Okay, so here's kind of the diagram of how it works. So carbon fixation into a 4C carbon molecule by Pep carboxylase enzyme in the mesophyll cell. So this top cell is the mesophyll cell, the bottom cell is the bundle sheath cell. So what's going to happen is we are going to separate the carbon fixation, which is going to happen up here, okay, from the actual Calvin cycle, which is going to happen down here. Okay, so typically they kind of run together, right? One big loop. So here, carbon dioxide comes in, and here is our PEP. I know it's a little bit small, but here's our Pep carboxylase right here. You can see the PEP right there. So Pep carboxylase is going to convert it into this 4-carbon molecule. You can count the black dots are the carbons. 1, 2, 3, 4. Right? There's 4 carbons right there. And now that 4-carbon molecule is going to go from the mesophyll down into the bundle sheath, and the Calvin cycle is going to happen away from the oxygen, right? Up here where their stomata is where the oxygen is going to be, right? There's going to be O2 up here, okay? And so you want to have the Calvin cycle getting away from the oxygen so that when you run the Calvin cycle, and I know you can't see it down here, but Rubisco is going to be down here, okay? So I'll just put RUB. Rubisco is going to be down here running the Calvin cycle, but your Rubisco is down here and your O2 is up here. So there's going to be very little photorespiration happening now because you separated where the carbon fixation takes place in the mesophyll versus the Calvin cycle taking place in the bundle sheath. And that's the whole idea of a C4 plant, and it does it by physically separating those processes. If we look at these plants called CAM plants, crassulacean acid metabolism plants, another plant that adapts to hot, dry climates. These are like your succulents, your cactus, your pineapple. They are going to separate carbon fixation from the Calvin cycle by time, not by physical separation, but by time. So they close their stomata during the day, and then they open their stomata at night. And so at night, all the carbon dioxide is coming in, right? Because the stomates are open, CO2 is rushing in, and they're taking that CO2 and they're converting it to also a 4C storage compound. And then during the day, when their stomata are closed, they're going to release that CO2. They're going to break down those 4C storage compounds, and that floods the whole compartment of CO2. And then that way, when they do the Calvin cycle, it doesn't have to compete with the oxygen anymore because there's so much CO2 in there that the concentration is so great that the Rubisco will bind the CO2 instead of the oxygen. Okay, so pretty ingenious little deal there. So if we look at C4 versus CAM, okay, both of them are trying to solve the gas exchange problem versus the water loss challenge in living in a hot environment. And so if you look at how these work, on the left you have your C4 plant, and you have them separating the carbon fixation and the Calvin cycle in 2 different cells, the mesophyll cell and the bundle sheath cell. So if we look here, right, mesophyll is going to be where carbon fixation takes place, and that's going to be with Pep carboxylase. And then down here in the bundle sheath, we're going to run the Calvin cycle away from the oxygen. Okay, and then over here, the CAM plants, they separate it by time, right? So they use 2 different times a day. So during the night, I know you can't hardly see that, but during the night, the CO2 is going to come in, and they're going to convert that into a C4 compound that they're going to store. And then during the day, they're going to break down that compound, and they're going to release tons of CO2, and they're going to flood this whole chamber full of CO2 that way, so that when you run the Calvin cycle, Rubisco will grab the CO2 instead of oxygen. Okay, so that's kind of pretty cool adaptations that plants have figured out over time. So that is photosynthesis. We're done with part 2. There's some practice questions for you, and good luck with all that. Let me know if you have any questions. See ya.